Hello, everyone. Today, it is my pleasure to introduce a METTL5-mediated 18S RNA m6A modification enhances ribosome assembly and ABA response in Arabidopsis. RNA modifications play central roles in influencing gene expression and cellular function, impacting various biological and physiological processes in eukaryotic organisms. RNA modifications occur on various RNA types, including mRNA and RNA. M6A has emerged as one of the most widespread and extensively studied modifications. Plans, the mechanisms involved in writing, removing, and interpreting the dynamic mRNA m6A methylation have been extensively examined in various development and environmental contexts, whereas knowledge on RNA m6A modification is rather limited. Highlights: Arabidopsis METTL5 specifically deposits m6A at a 1771 in 18S RNA. METTL5 enhances the translation efficiency of GSTs to mediate the ABA response. M6A1771 is indispensable for the binding of 18S RNA to rPL24A. In this study, we found that METTL5 mutants exhibited remarkably reduced M6A levels in total RNA and 18S RNA, while those in mRNA and 25S RNA were largely unaffected. M6A immunoprecipitation qPCR detected M6A methylation near the free foot end of 18S RNA, which was nearly abolished in METTL5. A modified METTL approach identified a C to T transition following a 1771 in 18S RNA, suggesting that a 1771 is modified. In vitro methylation assay confirmed that METTL5 exhibited methyltransferase activity toward this 18S RNA fragment. A catalytically inactive METTL5 variant showed greatly impaired catalytic activity on 18S RNA. METTL5 was required for stress response to ABA. Under ABA treatment, METTL5 mutants exhibited markedly decreased seed germination rates, green cotyledon rates, and seedling fresh weight compared to wild type. Furthermore, the ABA hypersensitivity phenotype of METTL5 were fully rescued by METTL5-free HA, but not by the TTL5-free HA construct. Translation activity analysis with sunset and polysome profiling indicated a notable reduction in global translation levels in METTL5 mutants compared to wild type under both mock and ABA conditions. Polysome-seq revealed reduced translation efficiency of 3,303 and 2013 transcripts in METTL5 compared to wild-type under mock and ABA treatment, respectively. Polysome-seq data revealed a clear decrease in the translation efficiencies of GSTs and GPXs, two gene families involved in detoxifying RS. qPCR further confirmed the significantly reduced translation efficiencies of free GST genes in METTL5 mutants compared to wild-type plants. A deficiency of detoxifying enzymes may lead to excessive accumulation of ROS, resulting in ABA hypersensitivity. DAB staining indicated METTL5 mutants accumulated significantly higher levels of ROS in root tips and leaves compared to wild type. Exogenous addition of GSH suppressed the growth inhibition caused by ABA in METTL5 plants. 18S RNA m6A1771 is located in the H44 loop of 18S, which interacts with the large ribosomal subunit protein rPL24. In vitro and in vivo RNA immunoprecipitation assays revealed that binding of rPL24A to 18S RNA was abolished in METTL5 mutants. Similar to METTL14, rPL24A was localized mainly in the cytoplasm. rPL24A mutants exhibited notable decreases in global translation levels, translation efficiency of GST genes and seed germination rates compared to wild-type plants. rPL24A mutation did not further enhance the ABA hypersensitive phenotypes of METTL5, supporting the notion that METTL5 and rPL24 act in the same genetic pathway in mediating ABA response. Together, METTL5 catalyzes the m6A methylation at a 1771 in 18S RNA, a modification essential for its association with the ribosomal protein rPL24, facilitating the assembly of 80S ribosome. This facilitates the translation of mRNAs encoding the GST, thereby maintaining normal ROS levels and ensuring proper ABA responses. In METTL5 mutants, the absence of M6A1771 compromises rPL24 incorporation and ribosome, impairing the translation of GSTs. This results in RS excessive accumulation and hypersensitivity to ABA. Summary: Arabidopsis METTL5 deposits M6A at a 1771 in 18S RNA to promote translation of GSTs to mediate the ABA response. Our findings uncover a mechanism by which RNA epitranscriptomic regulation controls mRNA translation and mediates plant responses to environmental conditions.